Thank you so much for giving me the opportunity to talk about the importance of adaptive sampling in nanopore long-read sequencing in pharmacogenetics. It's an honor for me to be here, and now I would like to share my disclosures, and I will follow this agenda. I'm going to introduce the basics of pharmacogenomics to be clear about the topic and to help you to understand why we need long-read sequencing and why we try this long-read sequencing in our hospital for pharmacogenetic analysis. So, a few words about the introduction, the history, the needs, and the challenges we have to face. First, I would like to just remind you that the pharmacogenomics was discovered by Pythagore in patients with J6PD deficiency. More recently, the definition was proposed by Friedrich Vogel. And in my talk, I'm going to focus on CYP2D6 and UGT1A1 gene. Why? Because they are key genes involved in pharmacogenomics, in pharmacokinetics and toxicity of drugs. So first, a word about medical pharmacology. In my routine use, we try to achieve the reference therapeutic range with a drug or a combination of drugs for efficacy at the reference standard posology. It works very well for most of the patients. Nevertheless, we have three situations. It happens in some cases that we do not observe any efficacy and any adverse events. In this case, we have to decipher, we have to determine the pharmacological mechanism involved to adjust the therapeutic. In other situations, we can observe some adverse events with pharmacological effect. In that case, we have also to identify the mechanism of toxicity for this patient to adjust the therapeutic. And the third situation in which, for some patients, we can observe adverse events, but no pharmacological benefit. So, for that, in my hospital, we decided to work on the prediction of the therapeutic response and the level of risk of toxicity using different drugs. So, we are working on how individual genetic variation can affect clinical response and the level of risk of side effects. So we focus on attention on some genes involved on pharmacokinetics, but also genes involved on pharmacodynamics. So why why we need this work? Just because several publications have demonstrated that the pharmacogenomic testing may make some benefit. It enables us to optimize treatments. It decreases hospitalization time for our patients and save money. Patients with pharmacogenetic testing to get their medication selection save money in our experience, especially for chronic diseases like in psychiatry. So, in my hospital, we organize weekly meetings between medical units, the pharmacy, and our department. We are in charge to the pharmacogenetics examination and titration of drugs. We try to make the most informed therapeutic decision possible for our patient. So, in the second part of my talk, I would like to remind you the basics of pharmacogenetics and what we are missing. So a few words about the CYP2D6 gene. It's a really important gene in pharmacogenetics because it's involved in the metabolism of numerous drugs. It's a small gene, but it has two pseudogenes. And the CYP2D7 is a very homologous gene, so we have to differentiate both of them. In addition, CYP2D6 gene can have a lot of polymorphism. Some of them are linked to non-functional or decreased function alleles. So we have to identify these variants. But we have additional variation to study. In some patients, we can observe some deletion of copies of the CYP2D6 gene, or we can also observe the duplication. And it can make some hybrids with the pseudogene CYP2D7. So currently in my hospital, we are verifying by orthogonal MLP techniques and other techniques all the structure forms. Now, a few words about haplotyping and star allele nomenclature. Haplotyping, it's a group of variants, and the star allele nomenclature is the common nomenclature we are using in pharmacogenomics. It's defined a genetic signature affecting how a drug is metabolized. Usually, star 1 is used to design the reference sequence. And from the diplotype, we can we can associate a metabolic activity score. It's really important in routine settings because according to the variant, the diplotype we determine, we can say whether or not the patient is a slow metabolizer, an intermediate metabolizer, or an ultra metabolizer. It can help us to determine the risk for a toxicity or a non-efficacy drug. It's very well described in this paper. So now, currently in my hospital, we developed a very nice, accurate tool with a Sophia Genetic company. It enables us to study the CYP2D6 and the CYP2D7. As you can see, we are able to count the copies of the both genes. Normally, we have 2 alleles. But in some cases, we have additional allele. It's a duplication. As you can see here, the number is 3 instead of 2 at the line of the CYP2D6. But it can make hybrids, and it can be combined. And as you can see here, we have an hybrid with the beginning of CYP2D6 and the end of CYP2D7. So the problem is with short-read sequencing, we cannot, without family members sequencing, determine the exact chromosome structure organization. So in my current activity, we have a delivery time variable according to the urgency. For example, when we want to prescribe thiopurine molecule, it cannot be done before the pharmacogenomic analysis of the TPMT gene because we have some variant associated with life-threatening. But for psychotropic agents, we have a little more time, and we can release the report within 7 to 10 days. But we have to study more genes. So now, in my last part of the talk, I'm going to talk about our experience on long-read sequencing for pharmacogenetics analysis. So, we selected 26 samples representative of genetical variation of CYP2D6 in our routine. We sequence the sample using our certified workflow. It's a short-read sequencing method associated with the bioinformatics from SOPHIA Genetic. It takes at least 5 to 10 days to release the result. In this experience, we sequence this same sample using Nanopore technology. We use the adaptive technology. And we were able to sequence 3 to 4 samples per flow cell, and it takes 24 to 70 hours to get the results. So now a few words about the concordance. As you can see, we have a great concordance. We of course, observe some discordances about the genotype, but it's just due to the statistical phasing we have to carry out using our short-read data. And the phenotype, the score of the metabolism, is not modified. For 2 cases, the Pangu public tool we use for the haplotyping was not able to detect the duplication of CYP2D6. It's the case 3 and the case 18. It's not a problem for the case 3 because STAR10 is not functional, so the phenotype is not modified. But I will talk about a little bit more on the case 18 because the duplication is a functional copy. Now, just to highlight the very great and perfect concordance on the SNV detection. We have a nice detection, and we may take the benefit of the direct phasing to highlight that we are able to see that star 29 is in one allele, while star 10 is in the other. So the heterozygosity is very nice and we have the true answer. Now, as I mentioned previously, that Pangu tool was not perfect for the detection of duplication. Oxford Nanopore company developed a nice and promising tool, Chinook. It is developed for comprehensive CYP2D6 analysis. It incorporates CYP2D6 and CYP2D7 allele reconstruction and enable us to get the CNV calling and the diplotype. It's really nice, as you can see on that alignment. It's the true answer, and it's more confident for us. So I would like just to highlight another example. In this example, we have a duplication of STAR2 and an hybrid, as you can see in this short data. But I'm not able, just with this data, to determine the configuration of each chromosomes, because we have duplication and hybrids with other copies. So, using a Pangu tool, but better with Chinook tool, we will be, in the future, able to differentiate these different copies and to identify also the exact position of the conversion site we have in the hybrid. It's a real improvement. And in this case, it will be very nicely highlighted because in my routine, using the information I had when I did this testing, I couldn't identify precisely the conversion site in this hybrid. It's in a genomic region with high homology between CYP2D6 and CYP2D7. So I indicated star 13. But Pangu tool indicate me it's star 79. So after analyzing, making the alignment, we can highlight the right position of the conversion site. And in fact, it's in exon 2. Now, I would like to finish on the comparison with other genes that we are studying in pharmacogenomics. We show a very great concordance between short and long read techniques. We can see here that ALDI public tool is more accurate in comparison to UR tool, but we do not have any discordances in terms of phenotype. Now, I would like just to finish on two small variations, two different variations. It's a small deletion in the promoter of UGT1A1. This protein is involved in the metabolism of the bilirubin, but also the metabolism of irinotecan, an anti-cancer drug. And as you can see here, we have to count the number of T repeat in its promoter at its location. And according to the number of T repeat, the expression level will be different, and the activity of metabolism, the metabolic activity will be different. So, as you can see in these three cases, it's really easy to determine this modification. Another modification we have to look at is larger deletion. Currently, I'm not able to identify this deletion using short read technique because it's too large, and we do not have the technique to do it correctly. As you can see here, we are able to detect the short deletion, 43 BP deletion in this promoter. And we can very nicely see the long form from the short form of this deletion. Now, I would like to finish with the key information on this comparison. We didn't observe any inferiority using long-read technique for the detection of variants accessible with short-read sequencing technique. We highlighted some improvements with long-read sequencing in terms of the detection of CNVs, in terms of the discrimination with the pseudogene, cytochrome 2D7, and the direct phasing is a very nice improvement to get the true answer for the haplotyping. Of course, it's nice to be able to visualize repeated region and large deletion. In the future, I think that the long-read sequencing approach will be nice for large gene analysis for which the current identified variants are not sufficient to explain phenotypes. And in the future, we will be able to analyze the methylation, and it will probably help us to explain the differences, the discordances we observe between phenotype and genotype. Because in pharmacogenomics, we are quantify in blood the concentration of the drug, but also their metabolites. Of course, we have some work to do. We have to work on ALDI algorithm, maybe on Pangu algorithm, to fix the errors we can observe in our cohort. We have to work on the visualization of the conversion site in the hybrid because it's important to be accurate. And I'm sure that the Chinook tool will be a real improvement in the field. So I would like to thank all the patients and my collaborators. I would like to thank Nanopore company for this project, and I have a special thanks for my team, medical team, diagnostic team, and bioinformatic team. Thank you so much for your attention.